Welcome to the Parson series, guys. This is your chapter number 3, part 1. So this is concerning the physiology of vision. So let us try to see some of the amazing concepts of Parsons regarding the physiology of the eye. Now they are saying that the function of the eye is to form a clear image of a given stimulus on the surface of retina. Now how it is going to form a clear image? So the anterior surface of the cornea accounts for about 2/3 of the refractive power of the eye. So this we already know that out of plus 60 diopter, cornea has plus 45 diopter, 43 to 45 diopter, that is your 2/3. While the lens refracts the light entering the eye and it is responsible for the ability to see objects at all the distances. So this actually responsibility is divided between the cornea and the lens. The cornea has the 2/3 of the power, lens has just 1/3 of the power. But yes, cornea is the most important refractive surface, being the anterior most, and I told you we have also the maximum difference of the refractive index at the anterior surface of the cornea. So the cornea is doing a great job. 2/3 of the power is actually given by the cornea, while the lens is helping to see the object at the near distance also and at a farther distance also, and this it is doing by changing its shape. Now you know, when we are bringing the objects nearer to the eye, the lens changes its shape. It is helping in accommodation. Similarly, when we are looking at a very distant object, then also lens change its shape. So we can say that we have an amazing autofocus lying in our eyes, just as you have got the cameras and they are focusing at each distance at which you want to click a pic. Similarly, up to some extent, like when it is very near, then also we have a problem, and when it is very far, so it has some limitations, but amongst the normal limit we can say that lens is able to do the autofocus. Okay. Now, this another important thing is your pupillary size. The pupillary size is adjusted for varying light intensities by a reflex contraction as well as the dilatation of the muscles, that is your sphincter pupillae muscle and the dilator pupillae muscle. Now, there is an amazing, balance between the two pathways, the parasympathetic and the sympathetic, which is helping to regulate the size of the pupil. Whenever there are dark conditions, we already have light, then is the parasympathetic pathway working, due to which we have constriction of the muscles and the pupillary size will be small. Similarly, if we are going in the dark conditions where the conditions are very poor, the illumination is very poor, now I require a larger pupil. So in that case, sympathetic pathway will be working, and due to the sympathetic system, we will have dilator pupillae dilating the pupil. So they are talking about the power. We have cornea, we have lens, we have autofocus, and then we have the pupillary reflex. Now going ahead, we have 1 million retinal ganglionic cells, and specifically speaking, it is 1.2 million, right? We have got 1.2 million ganglionic cells present, and as against these 1 million retinal ganglionic cells, they are saying we have 125 million photoreceptors. Now, why this is important? Now, because you do not have a balance, you have got 1 million retinal ganglionic cells while you have 125 million photoreceptors. At some places, you will have the good coordination between the two, while on the same time at some places we will not have that much good coordination because both the cells are not same in number. So this is why what is happening, if you look at the foveal region, in the foveal region we have what you called as 1-to-1 correspondence between the two. Now this is again amazing concept, because we know that fovea is the most sensitive part of the retina. The sharpest image of the object will be formed on the retina. Why? The reason is there, we have a good amazing 1-to-1 correspondence between the photoreceptors and your ganglionic cells. But as you go towards the periphery, we have more photoreceptors and therefore there will be loss in resolution. Now specifically speaking, more photoreceptors in the periphery, they are not just the photoreceptors, they are the rods, because cones are more in the central area, in the central fundus. So I will say rods are more in the periphery. Okay. Now, if you look at the next one, the eyes are converting the light energy which is lying in the wavelength of 397 to 723 nanometer into the electrical signals into the optic nerve. Now, this is again a practical concept. Whenever the light is entering the eye, it is passing through cornea, the aqueous humor, the lens, the vitreous humor and retina. Up till that time, it is the light wave only. But after some time, when it has to pass from the retina onwards, it has to be changed into the electrical impulse, because that electrical impulse will be taken up by the optic nerve. It will be going into the visual pathway, passing through the optic chiasma, optic tract, lateral geniculate body, optic radiations, and finally it is going to the striate cortex. So for that we require it in the electrical form. Now, if I talk about a single rod is able to detect a single photon of light. So there we have a one-to-one relationship, but problem is that the rods are low resolution. So because rods do not have that high resolution power, that is why the image that is formed is very blurred. It's not very good quality, though it has one-to-one relationship with the photon of the light, but again resolution quality is bad. Okay. Now coming to the optic disc, they are saying that the rays which are falling on the optic disc are giving no visual sensations, and that is the reason it is called as the blind spot of Marriott. Now why it is taking place? See, if I talk about the posterior surface, it is something like this. On the nasal side, this optic disc is there and that is continuing to form the optic nerve. So if you see specifically, this area is your optic disc and we have a vacuum, because you know the nerve fiber layer is continuing something like this and it is forming the optic nerve. So if I talk about this area, okay, this area, if the light is falling, then it will not form any image, and that is the reason that we have a blind spot there in the corresponding temporal field. So that is called as the blind spot of the Marriott. Alright. Now, the light which is falling upon the retina has the two essential reactions. We have a photochemical reactions and we have a electrical reactions. We will be looking at them one by one. Okay. Now, first if I talk about these photochemical reactions, okay, the photo and chemical. So, light induced reactions in the pigments which are taking place, and these pigments are present in the rods and cones. That is your photochemical reaction, and you will see that is called as the phototransduction. Now, the most fully explored pigment is the rhodopsin pigment. Rhodopsin pigment is also called as the visual purple. The most fully explored pigment about which you know much things is your rhodopsin, that is called as a visual purple, and this is obviously found in the rods because it is rhodopsin. Now, several related pigments have been discovered in the rods, while it seems that we have got three different pigments associated with the foveal cones. So, mainly we have the rhodopsin in the rods, while we have got three pigments in the cones. We will be talking about the rhodopsin here. Rhodopsin is actually a chromoprotein. Chromoprotein means a part which is present here is the chromophore. So, this chromophore is responsible for the preferential absorption of the light attached to a protein, and that is acting as a support. So, if I talk about the rhodopsin, rhodopsin is a chromoprotein. Chromoprotein, chromo means the chromophore, and chromophore means that it is a preferential absorption of the light attached to a protein. Now, this chromophore is actually belonging to a family of carotenoids. So, what is the property of this carotenoids? When exposed to the light, it is broken down through several intermediaries to the colorless vitamin A. So, this you know, through multiple sequence where you have got number of intermediates are formed, ultimately it is changing to the colorless vitamin A. and that is why it is said that vitamin A is good for your eyes, carrots are also good for your eyes, and this reaction is actually reversible. Now this is again an important thing, that so many steps are taking place, but ultimately this reaction is reversible. It is a photochemical reaction which initiates the visual process, give changes in the electrical potential and transmit it through. Now you have already talked about the rods and cones, which were your photoreceptors, and now you also know these are your first order neurons. Okay, so from the first order, where it will go? So that is your common sense. See, they are saying that they will go to the bipolar cells and then it is going to the ganglionic cells. So this is your second order and this is your third order. So by yourself also you can imagine how it is going, because now you know that these are the end organs. They are also the organs where the receptor potential is generating. So they are first order, and from this it will go to second order and third order. So they are going to visit your bipolar cells and also the ganglionic cells along the fibers of the optic nerve to the brain, and ultimately along this optic nerve they are going into the brain. They contain the pigments which is composed of opsin. Now another important thing that you should know is your opsin pigment. This is your opsin apoprotein. Okay, now the pigments which are present in the cones, you do not know much about it. So we know about the rhodopsin that I have told you, but it is likely that each preferentially binds to different bands of wavelength and they are seen as red, green and blue. So we do not know much detail about the cones and their pigments, their working, but the thing is that we have got some part which is reacting to the specific wavelength, and that is why we have got different cones like red color, green color and the blue color, right? Now coming to the rhodopsin cycle. Now as I already told you that rhodopsin is the visual pigment which is present in the outer segment of the rods. Now again this is an important question. Rhodopsin is present in the rods. Which part of the rods? The outer segment, and it is actually responsible for the scotopic vision, that is ability to see in the dark. So rhodopsin is present in the rods. Rods help in the darker vision, so this will help in the scotopic vision, and this rhodopsin has 11 cis retinal as the chromophore. Now what is happening? When this rhodopsin has this 11 cis retinal, it is embedded by 3 different chemical bonds. Okay, inside a single peptide transmembrane protein called as opsin. So we have got rhodopsin. So opsin is the apoprotein which is present, that is your transmembrane protein also, and the role of rhodopsin in the signal transduction cascade of vision is to activate the transducin. Now when this phototransduction, the signal transduction is taking place, what is actually the role of this rhodopsin? It is to activate the transducin. Okay, very important. And what is this transducin? It is a heterotrimeric G protein upon the absorption of light. Now can you see the catch point? The role of rhodopsin is to activate the transducin, and transducin is actually a G protein. So what it is doing when it absorbs the light, it is activating the transducin, and that is the reason why this is called as phototransduction. And this is the reason why this is actually called as the phototransduction. Now let's see ahead. The absorption of the of a photon of light by the chromophore leads to initial photoreaction, and then we have got conformational changes of protein that results in the activation of transducin. So we have got a number of photochemical reactions which are taking place, due to which we have got the activation of this transducin. And what will happen? The rhodopsin is activating the transducin, and while activating this, it is losing its color. And that is why we have got the term bleaching. Bleaching means the losing of color. So while doing the phototransduction, while activating this transducin, the rhodopsin is losing its color, and that is why it is called as a bleaching. And the cis trans isomerization of the chromophore is also occurring during this bleaching. So let us see the picture in which they have shown. Now can you see this? This is your rhodopsin, and the light energy is falling here. So there are number of intermediates which are forming. You have got barthorodopsin, lumirhodopsin, metarhodopsin, and you also have metarhodopsin. 2. We have got meta-rhodopsin and you also have the meta-rhodopsin 2. Now what will happen? This will be giving you two parts, the scotopsin and you have got the 11-trans retinal. Now they will be changing interchangeably in the 11-trans retinal and the 11-trans retinol, and when the enzyme isomerase will be acting on them, they will be forming the cis form. So we have 11-cis retinal and 11-cis retinol. This is your rhodopsin cycle. I think you must have read this in your physiology also. Now this is the time where we are actually trying to integrate with the help of this Parson series your physiology, your biochemistry, your pathology and your ophthalmology also, because that time when you read this rhodopsin cycle in the physiology, you do not have any idea about the ophthalmology that time. Now by the time you reach to the ophthalmology, you have already forgotten your rhodopsin cycle. So this is an attempt to bring the two things together. Okay, now what is happening? 11-cis retinal and 11-cis retinol, okay, they are now combining with the opsin protein, and when they combine here with the opsin, they will be forming the rhodopsin again. So this is your visual cycle. This is your visual cycle or the rhodopsin cycle. I hope this is very, very clear. This we have covered in your the notes also. Now, if I talk about your rhodopsin, what they are saying, the formation of the electronically excited state of rhodopsin and the photorhodopsin as well as hypsorhodopsin has been observed almost immediately. These are formed. The parthorodopsin is formed in picoseconds, then is the lumirhodopsin, then is the then we have metarodopsin 1 and then you have metarodopsin 2. So actually the intermediates are formed in the very, very small fractions in the picoseconds, milliseconds, microseconds, and ultimately a stable product is forming. Okay. Now a human rod cell can respond to absorption of a single photon, demonstrating the efficiency of the photoisomerization, a very important thing. Can you see they are saying that the human rod cell has that much potency that it can actually respond to even a single photon, that much is its potency. So I hope this concept is very, very clear. Now we come to your magnocellular, parvocellular and the coniocellular pathways. So when the photoreceptors are stimulated, okay, you know that these are your first order neurons. They are transmitting the signals to the bipolar cells, that is your second order. Then they are connecting with the retinal ganglionic cells, that is also third order. So this is easy to understand, okay, very simple one. Now, the retinal physiology at the level of the neurotransmission and communication between the cells is quite complex, with the cells connecting in specific patterns depending on the nature of the stimulus. For example, some domains are activated when a light is switched on, okay, and others when the light is turned off. So, in order to make it happen that every time you are doing something, they are activated. So, we have made them, we means so nature has made them in such a way that some are working when the light is on and some are also working when the light is off, because I require the working 24/7. So in addition, the retinal ganglionic cells themselves are of three types. In human retina, one class of ganglionic cells are smaller and have thinner axons of smaller caliber, and that one is predominant in the macular region. These ganglionic cells are actually color sensitive, and that makes sense also because these ganglionic cells are present in the macular area. So obviously they have to be color sensitive. The physiological response is of the color opponent type with the high special frequency. These are called as the P cells and form the part of the parvocellular system. So you already know that certain kind of cells which are small size are present in the parvocellular pathway. And these parvocellular ones are actually layers 3 to 6 of the lateral geniculate body. So these are your small size cells and they are responsible for the very, very fine details. So here also that means high special frequency is required. Now these P cells responds transiently to constant stimulation with short latencies and also is color selective. So I always tell you that it is actually concerning the fine details, the color, the texture, the contrast. So the same thing they are saying. Okay. Now the axons of the P cells, which constitute about 90% of the retinal ganglionic cells, terminate in the parvocellular layers of the lateral geniculate body. So P you can remember for parvocellular. So these P cells, which are high special cells responsible for the color sensitivity, are terminating in the parvocellular layer of your parvocellular layer or parvocellular pathway of the lateral geniculate body. The remaining are your M cells. M cells will be corresponding to your magnocellular pathway, and these are present in your layers 1 and 2, which are the containing the larger cells. So these are your larger cells, they are thicker axons, they are having high caliber, and they are fast conducting. So they are transmitting high temporal motion related information of low spatial frequency unrelated to color. So this is actually responsible for the mainly movement, depth and flickering and all. So high spatial frequency is actually related to what you called as the parvocellular low spatial frequency. If you see the low spatial frequency, they are located in the magnocellular and they are mainly concerned with the fast conducting and high temporal motion. Motion related information is going through this magnocellular, right? The lateral geniculate body regulates the neural information from the retina and then it is going to the cortex. It organizes the information in different layers. So you have got the different layers, like for example, the ipsilateral input to layers 2, 3, 5 and the contralateral to 1, 4, 6. So this information you already have. We have got the ipsilateral input in certain layers from the same side. Ipsilateral means the same side. See, lateral geniculate body, you know, have got 6 layers. Lateral geniculate body has got the 6 layers, that is your 1 to 6, and out of these 6 layers you can divide them into 2, 3, 5 as well as the 1, 4, 6. So 2, 3, 5 they will be going in the receiving the input from the same side, while the 1, 4, 6 is contralateral, that is your opposite side. So this also you know. Now the retinal cells with a particular function project to similar lateral geniculate nucleus cells. For example, those with red on, green off center around the receptor fields. They are saying that you have got so many layers in the lateral geniculate body having different kind of cells. So those all which have got similar kind of function will be present in a particular layer nearby each other. So if I talk about the M cells were synapsing in the magnocellular layers, they are 1 and 2. P cells in the synapse in the 3 to 6 of the lateral geniculate body, they are the parvocellular, right? While what about your conio cells? Conio cells are present in the intralaminar area. Conio cells are present in the intralaminar area. Now, the cortical visual system is thought to consist of 2 areas. We have got the primary visual cortex. See, this is your neuroanatomy. Again, we are integrating here. So, the primary visual cortex is your area 1 that transforms the information received from the lateral geniculate body and it is sending to the different domains. And also to the secondary center. So, these secondary or the extra striate cortex is V2 and this is for the transmission to the higher visual area. So, you have got primary area and you have got secondary areas of vision. It is likely that the visual attributes of color, form, motion are not neatly segregated. Instead, there are just 2 main streams composed of magno, parvo and conio geniculate cells. They are saying that we have got so many things, like we have got color also, we have got form, we have motion, but there is no strict segregation of all these things, but we have got broad segregations of 3 things. We have magnocellular pathway, we have got parvocellular pathway and you have got the coniocellular pathway which is lying in the intralaminar area. So magno means large, mainly in 1 and 2, you have got M cells, these are low spatial, high movement. Then we have got the parvocellular P cells, these are small cells, they are lying in the layers 3 to 6, high spatial, and they are mainly concerning about the color sensitivity, the small details, and in the intralaminar area we have the conio cells. So that's all with this part. I hope we have got so many high yield things in this part: the visual cycle, rhodopsin, then the one-to-one relationship between the rods and cones, between the ganglionic cells and the photoreceptors, why fovea is so sensitive, along with your different pathways in the lateral geniculate body. See you in the next part. Thank you and happy ophthalmology.